Right. Okay, everyone. Let's see. Yesterday we did the biological test for Enterobacteriaceae. Dr. Bailey and I did. So I'm going to make a couple of really short videos and show you the outcome so far. So it literally was in the incubator for about 24 hours. I think it would be exactly just about 24 hours. So we got some really good results. So the timing is perfect because the cultures were about 24 hours old. We inoculated them, then we waited 24 hours in the incubator. So these were all new cells. So you know that during, well, hopefully you know that with the younger cultures, they're going through a lot of mitotic division because we culture them on fresh plates and we culture them in fresh tubes that were at room temperature, so they didn't have any competition. They were very, very young, reproducing very, very quickly. So things worked out really well. So I just wanted to, hopefully this will work. So I'm on MacBook. So literally that is, you can probably see that. This is a white background there. So that is the MRVP, methyl red Volks-Park-Hour test. We're going to divide this in two and add some methyl red, which is a pH indicator, and Volks-Park-Hour solution A and B, and we're going to look for a different result. So with the MRVP, I'm going to show you the different vials, and you'll see that the turbidity is different. So that tells us for sure that something's growing. Now I can't obviously tell you that everything in there is 100% alive. They'll tell me the turgor. So if I shine light through it and it's denser, I know that something's growing in it. But you know, with microbacteria, that we can't literally take their blood pressure and respiration because we don't know if they're alive. We could plate them and see if they're growing. I'm assuming they're all alive. It's only been 24 hours. So we're going to do methyl red, which is a pH indicator. So a lot of these, this contains glucose, and what's going to happen is it's going to change color as the microbes start fermenting the indicator. They're going to change the pH lower. So it's going to be red, telling us that the pH is lower, and because it's fermenting glucose, producing an acid, which obviously will drop the pH. Where we can use the Volks-Park-Hour test, and that will turn yellow. It's going to, after it consumes glucose, we're going to be checking acetyl carbonyl is indicated by the presence of pink color after the additions of alpha naphthol, Volks-Power A, potassium hydroxide, Volks-Power B. And then you guys aren't here, so I tell you, potassium hydroxide is very, very caustic. So I'll be doing the test here, and we'll either, I'll probably snapshot the pictures and we'll upload them for you. Yeah, so we'll check for that color. All right, so that is an uncultured tube. That's how we started. That's our baseline. All right, so I'll hold this up too. So if you look, this is baseline. This is E. coli. All right, and we know E. coli regenerates about, or goes through mitotic division about every 20 minutes, logarithmically. So we can assume that there's a lot in there. This is Pseudomonas. Wait a second. Oh, I'm sorry. Yeah, that's Pseudomonas. Different. Proteus. That's Proteus. You can see the difference there. Serratia. See, there's turbidity in there too. Klebsiella. All right. There's another one of E. coli again. Inoculated a little bit later, so it's not quite as turbid as the original. All right, I am going to pause this and I will label this. So this is the MRVP. And what I'm going to do is I'm going to divide these in two. we'll do methyl red MR, and then Volks-Krakauer for the other part, checking for fermentation of glucose to change the pH. And then with the Volks-Krakauer, we're going to do, it's going to ferment the glucose. After that, we're going to be checking for acetyl methyl carbonyl. If it produces that, it's going to turn it like a yellowish pink color, right? Because we can see it's this color now. It'll be a yellowish pink with Volks-Krakauer, and with methyl red, it's going to be more red. And it's in your lab sheet. It's right on here in your lab sheet. All right.